Assalamualaikum warahmatullahi wabarakatuh. My name is Rena Juwita BPT 0922222004. So today I'm going to present my slide is from the title is the From Lab to Clinic, the translating nanomedicine into commercial success. And with this topic, I'm going to share a story about the journey of doxorubicin. Let's start with a simple idea in a nanoparticle. They come in a different shapes, size, and even personalities. Some are soft and some may be hard. The use of the nanoscale structure for medicine isn't actually new. Liposome and the dendrimers were already around more than 30 years ago, and some are already in a clinical use for nowadays. But the real leap in a nanomedicine in this 21st century is about making these particles smarter, such as able to target specific tissue, control drug release, and avoid being cleared too quickly by the body. And one of the biggest advantage is in the cancer treatment by designing drugs at the nanoscale, so we can target the tumor cell more precisely, and we also can reduce the side effect. For example, the chemotherapy drugs, doxorubicin, is effectively but highly toxic. So it is especially to the heart. So by putting it into liposome form, we can change how it moves in the body by reduce toxicity and still keep it cancer fighting power. In this presentation, we will focus on the doxil. It is the first FDA approved nanomedicine. So we'll see how it moved from the promising lab invention to a commercially successful drug, cancer drug. And the quality control and the regulation along the way. This is the scale up. Turning a lab discovery into a large scale medicine isn't as simple as just make it more of it. For Doxil, one biggest challenge was controlling particle size. Size affects how the drug travels in the body, how well it reaches tumors, and how quickly it releases. Even small changes in the process could cause a big difference in the results. To solve this, the team of the medicines used microfluidics technology, a method that precisely control particle size and keep it consistently even when the production is scaled up. So another challenge was keeping the liposome stable. The temperature had to be careful controlled because too much heat or uneven mixing could damage the liposome or cause the drug to leak out. And finally, there was a problem of a batch-to-batch uniformity. Traditional batch manufacture can be slow and inconsistent. By switching to continuous production method, they could make a doxil more reliable with the same quality every time. Next is the CGMP, current good manufacturing process in another medicine. Okay, so now, even if you can make doxil at a scale, it still has to meet a current good manufacturing practice or CGMP. This means every step of production must guarantee quality, safety, and consistency. For liposomal drugs like doxil, important factors include temperature, pH, particle size, and endotoxin level. The temperature affects the structure and the drug holding ability of the liposomes. For pH, that can influence stability and the drugs release, especially for smart stimuli responsive designs. The particle size determine how the drugs behave in the body. Too big or too small, and it won't work as intended. And the endotoxin must be kept extremely low because they can trigger harmful immune reaction. So on top of that, the manufacturing need to happen in sterile, specialized facilities. This avoids contamination and ensure every batch meets the same high standard. Next is the regulatory challenge. It's a getting FDA approval for Doxil was not a small task. It combined three novel features: pegylated liposome to extend circulation time, a spatial loading method to track deoxyribonucleic inside, and a lipid bilayer designed for stability. PEG or polyethylene glycol was thought to be safe and non-immunogenic, but later research found that the PEG is attached to another molecule like lipids. It can sometimes trigger immune responses. This raised concerns about the safety and meant the developers had to run extensively preclinical and clinical studies to prove their formulation was safe and effective. It shows that with innovative drug delivery systems, you not only have to prove the drug works, but also that the delivery method itself is safe. So the conclusion for the presentation is Doxil was a breakthrough, the first liposomal drug to make it from lab to clinic by changing the drug cancer treatment with a better target and a fewer side effects. Its success wasn't just about the science of pegylated liposomes, it was also about the solving reaction world problem by scaling up production, ensuring quality, and meeting tough regulatory demands. Doxil paved the way for future nanomedicine influence approval pathway and proving that with a strong collaboration and innovation, even the most complex idea in the lab can become life-changing treatment for the patient.